Alright guys, welcome back to another A to Z explained AP Bio videos. So today we're going to be talking about lipids. Okay, so basically what are lipids? There's no really good term for this, but basically it's just any molecule that's hydrophobic or amphipathic. That's a mouthful. And yeah, you probably guessed it, hydrophobic means that it doesn't like water, and amphipathic means like half of it likes water, half it doesn't like water. So let's kind of just take a look at some examples of these. First off, we have monomers. So you have things like glycerol, and that looks like this. And things like fatty acids. So this is a glycerol and fatty acids. And those look like this. All right, so that's glycerol. That's a fatty acid. Those are pretty simple. Basically, they're hydrophobic because they really don't get along with water, and they really just repel water. So anytime you think of like something hydrophobic, really be thinking, okay, lipids. So what's the function of a lipid? And because it's a super diverse category, there's a ton of different functions. So first of all, we have energy storage. So those are things like your fats and your oils. So that makes sense, right? Like butter, olive oil. We use those for energy, and those are lipids, right? You can't mix oil and water, right? Because it's hydrophobic. So that's like one example. You have things like structure, supporting the structure of your cells, and you have things like signaling, and that is like sending signals within your body. So there's so many different functions of lipids. You really can't know them all, but like if you know these three, at least you're gonna be pretty good. They don't directly test like, oh, do you know like what lipids do, but really just building off of that knowledge. So now we can talk about some polymers. So let's talk about triglycerides. And you guessed it, tri being 3 and glyceride being glycerol. And so this looks a little something like this. Okay, so this is a triglyceride. And so basically, this diagram isn't great because it's kind of tight to write it. Also, anytime you see CH2, it's just a carbon with two hydrogens bond to it like that, but it's easier to write it because it saves space. So basically, as you guessed it by the name, you have this, glycerol, modified glycerol molecule up here, and then you have these 3 fatty acid tails down here. And so this is really important. This is a really good type of fat, and it also can be used to demonstrate a lot of different things. So basically, this can talk about saturated and unsaturated fat. So I'm just going to draw like a separate diagram. I'm just going to draw it like this to save room. So those are the first two, like these two being like this stuff, and then this top bit being like this stuff. And then this comes down, same thing, and then I'll show you guys what I mean. So you're down, you have like a CH2, and then you have something special, because what this looks like is goes down to a C, H, and then the double bond, I'm going to show this in red here, down to a C with an H, and then back up CH2, and then CH3. CH3 is just this ending molecule there. So what happened here is because of this double bond, that actually caused the fatty acid tail to bend. And so we call this an unsaturated fat. And so because this has one bend, right, this bend right here in the shape, we call this a monounsaturated fat. But then we can go into polyunsaturated fats. So applying this concept of this double bond makes it bend, I'm just going to draw the shape because the actual molecules, all you need to know is that it's caused by double bonds. But we have a polyunsaturated fat in 2 different ways. You can have 2 bends on two or more, but let's say you have 2 bends on this, right? So you have one bend here, right? So that's a polyunsaturated fat, again, caused by double bonding. But you can also have two bends on the same tail. And so that would look something like this. We have that first bend and then a second bend. So these are both polyunsaturated fats. But this does have a really important application because it can tell you a lot about whether it's solid or liquid at room temperature. So let's take a look. This is an example. But let's say we have like a cup, right? Okay, so let's say we have this cup here, right? And let's say we fill it with bacon grease, right? And what do we know about bacon grease at room temperature is that it's actually solid. So the reason this happens is because bacon is it's a saturated fat. And so what this looks like, if you look on the molecular level, it's a bunch of these little structures. Okay, so we have all these structures, and this is really similar to the reason why ice is less dense. But basically, at room temperature, we have all these things, and because they're so uniform, right? They're saturated, so all their fatty acid tails are straight. You can actually pack them together really tightly, and that makes it really easy to become a solid at room temperature. Or we have this other example with unsaturated fats, where if we have these tails, right, then these things are all out of whack. And it's going to be hard to stack them all together because they aren't the same shape, so they're just bouncing around, right? It's like, imagine stacking like a bunch of squares in a box versus stacking a bunch of random Lego pieces in a box, and they're just bouncing around because you have like the circle ones and the square ones and the long ones and the short ones. And so this is why unsaturated fats like olive oil is liquid at room temperature, because it doesn't have the right shape to compact together. Now, time for probably one of the most important things in AP Bio. It's the phospholipid. And I know I was talking about water being like the biggest thing in bio. This is probably the second biggest thing in bio. So what this is it's, as you guessed it, phospho being phosphate and lipid being lipid. And this is so big. If you're going to learn one thing from this video, or maybe even this unit as a whole, it'd be this. But basically, it's a phosphate group with two fatty acid tails, and it looks something like this. Okay, so there we have one phospholipid molecule. And you might be looking at, oh, well, we have this pink thing up here, this green thing down here, and this black thing. And you'd be thinking, oh, why are these colors? But that's because this is an amphipathic molecule. And so basically what happens is you have this side, right? And this side is hydrophilic. This side loves water. It's a superpolar molecule. We have phosphate. We have a ton of oxygens. This side loves water. And this side, we have all this stuff, right? And this side is a hydrophobic molecule because it's a lipid. It's a fatty acid chain. So this is hydrophobic. So we have all these things, and what this does is this allows you to make the phospholipid bilayer. And this is like anything about cells and the way you work, it comes down to this thing. This thing is so important. And the way it works is I'm going to just use like this little scheme here. I'm just going to draw it like this. So this represents like the phosphate head. This represents like the fatty acid tails, just so we're good with that. You take a bunch of these phospholipids, and then you put them into a line, and what you do is you flip them. And this happens, right, because when you put it in water, you have these dudes that love water. I'm going to draw some water. These dudes love water. Water's like, oh my god, yes, polar molecules, awesome. Then these dudes, they hate water. They're like, no, get it away. And so what happens is these things get forced together, and they make this wall-looking thing, right? You have water lovers on the outsides and water haters on the inside. And so this is what cell membranes are made out of. Membranes in any like cell or any like subcellular molecule, they're all made of this stuff. This is so important when it comes to like cellular biology. This is the most important thing. So it makes a little wall. And so this is so important. This is basically the biggest thing. And you have these non-charged, non-polar molecules down here, and you have these polar molecules on the outside. And that's basically what lets you to have watertight cells, where you have water on one side, water on the other, and they can't pass through without other things. And then finally, we have some just other kind of assorted things where we have steroids. And these aren't super important. They don't come up as much as phospholipid bilayers, but it is important to know. So steroids are used for like signaling, and they have like this ring structure. It looks like this, right? It kind of looks like that. It's not super important, but you have these things like cholesterol, and so like that's used for like cell membrane support. When you have like sex hormones like estrogen, testosterone. And that's about it for lipids. Phospholipids, know this. If you're going to know anything, make sure it's phospholipid by phospholipids. You have hydrophilic on one side, hydrophobic on the other. This is so important. Anything with cells, it all really comes down to this. And then, know the structure of these, like monounsaturated, polyunsaturated, and just saturated. This is saturated fat. I didn't write it down, but just you have 3 straight dudes, saturated. And then, there's so many functions. Lipids are a super diverse molecule. So you have energy storage, you have structure, like cell structure, so that's cholesterol like we were talking, and cell signaling. You're going to learn a lot about this in Unit 4. But that's about it for lipids, and I hope to see you guys in the next video.